So the first speaker is Teresa. We'll talk about computational approaches to study interaction between mutagenic processes and cellular processes.
Okay, thank you, Robert. And I'll try to start by thanking the organizers, so Robert and others, for a kind invitation to participate in this workshop. It's been a really honor. It was wonderful to listen to all this talk. So I hope my talk will live to the expectations here and be as great as all the other presentations. So for the last couple of years, my group has been working on understanding of mutagenic processes, mostly in cancer, but also by extension, mutagenic processes also with application to other diseases. So okay, so as you know, a DNA in every individual cell is subject to constant damage. This damage can be imposed by just exposure to naturally, existing environmental factors. This damage may be also related to stochasticity and error-prone processes within the cells, the errors related to replications. And in fact, it's been estimated that on average, a cell can accumulate, not accumulate, but can be subject of about 10 to the 5 lesions per cell per day. So fortunately, most of those lesions are very efficiently repaired by DNA repair processes that a cell developed, and only a small number of those lesions leak out, remain unrepaired, and they will lead to mutations. As I mentioned, this process is very efficient, and those the number of those somatic mutations remains to be very small. But unless the amount of DNA damage is really high, or perhaps in some cases when the DNA repair is not as efficient as it's expected to be, this accumulation may be much more significant and eventually leading to cancer. So recently, the studies related to understanding of those mutagenic processes in cancer, but also in other contexts, have received a wonderful boost thanks to study of Alexandrov et al. So it has been already appreciated for a long time that certain mutagenic processes tend to leave characteristic mutational patterns into human genomes. For example, UV light is known to preferentially mutate C to T. However, those probability of this mutation of C to T is context dependent, and it's particularly present when we have where C is preceded by T. So those are basically T to C to T to T mutation. UV light is not the only process that has this type of characteristic mutation pattern. And one of the important contributions of the study of Alexandrov et al. Is to propose that we should really look and have a general framework, a common way of representing those mutational processes, those characteristic patterns that we are going to refer now as mutational signatures. So here I will follow the definition of mutational signature as proposed in the study. And basically the idea is that we are going to consider mutational categories. So for this study, the mutation categories will be a triple of nucleotides, where the central nucleotide is the one that's mutated, and 2 additional nucleotides are provided for the context. So this leads to 96 mutation categories. Up there you have the summary of the mutation of the central residuum, so C2A, C2G, C2T, and down here that you cannot quite see are those all this possible combination of the flanking residuum. So once we, with this formalism, we can look at some other known mutagens that are known to have characteristic mutation patterns. So, for example, the enzyme ApoBec enzyme that's known to mutate single-stranded DNA has again characteristic mutation pattern is again C2C, but the context is quite different. Some of the chemicals in cigarette smoking have yet another pattern, and various drugs might have the individual mutation patterns. So, of course, currently the number of various agents that may contribute such mutation signatures is unknown, but looking from the other end, starting from the definition, this general definition of mutational signature, we can ask the question, what are the mutational signatures that are needed to explain mutations in, say, cancer genomes that are available to us? So to answer this question, again, this is Alexandrov proposed to just simply use the non-negative matrix factorization, starting with the data that takes all the samples of available cancer samples, and then starting with this count matrix, where each column corresponds to one of this, each of those 96 mutational categories, and then counting for every individual, we count how many mutations in each category we have. So we have this count matrix, and the question is, identify those mutational signatures defined by those profiles, probability profiles I show on the previous slide, so that all those mutation counts can be explained as linear combination of those signatures. So we are, as I say, we are going to refer to those signatures as cosmic signatures, since they are being deposited in cosmic database. And one more thing is, since basically what we are having here is we represent all the mutation in the genome as linear combination of those signatures. So the coefficient of this linear combination for a given signature will be referred to exposure, and is a good terminology, because, for example, looking at the smoking signatures, the signatures that's associated with smoke, we can see that really exposure and the amount of the mutations that's attributed to the signature is related to the year back cigarettes, so related to how much a given individual smoked. So it makes sense to think about this coefficient as the strength of the mutational process. Okay. Oh. Okay. So following again the same studies, applying first time applying this non-negative matrix factorization in 2013 to the cancer genomes that were available at this point. The author identified 30 different cosmic signatures. Subsequent, a later study, 2020, with a much bigger cohort, came up with a much larger set of signatures. So here are some examples of those signatures as I identify here by numbers. And of course, we'd like to now understand what's the relation between those computational derived signatures and mutagenic processes in cancer. And some of those signatures could be immediately recognized and well understood. And for some of them, we can only propose some vague relationship. So for example, there are 3 signatures known to be we call it clock-like signatures that seem to be increased with the patient's age. Out of those, signature 1 is well understood, is related to methylcellulose deamination. So this process that happens naturally during replication. So the more replication cycle, the bigger accumulation of the signature you may have, but the remaining 2 are more vague. And then again, going through that list, some of the signatures are maybe understood. So for example, signature 4 is related to smoking, but still doesn't tell us exactly what the relation is, what is the mechanistic relation to smoking. And then many of those are really DNA signatures 11, DNA alkylating agents, but what signature 11 and not some other pattern is still very interesting to know. So how we are going to now go around and try to understand deeper those, the relations of those mutational patterns that we discover with the specific mutagenic processes that may occur in cancer. First of all, why do we care? Obviously, answering a question, this is very basic questions, always interesting. Would like to understand the cancerogenesis in general, but it's also important from the perspective more direct application to understand, say, for prevention, if we can identify certain mutation signature and link it to some experimental exposures, then we understand so much, and we know that the individual should avoid this exposure since it's mutagenic. But also, it's also important for therapy. So some of those mutagenic processes are related with deficiency of DNA repair machinery. That, for example, in breast cancer, homologous recombination deficiency is very, very abundant. So if we understand the DNA repair deficiency associated with given signatures, then this may propose, what is the complementary pathways that the cancer cell use to overcome, and by using a synthetic lethality, hitting this complementary pathway may selectively kill the cancer cell. So this is in fact the idea behind PRP inhibitors being applied to breast cancer with homologous recombination deficiency, but also later on, once been appreciated that homologous recombination deficiency is associated with specific signature, this therapy could be also brought to, say, also ovarian cancer that had that signatures, and therefore suggesting that's homologous recombination and so on. There are other signatures that are associated, again, work well with immunotherapy and so on. So it's really understanding what are the signatures and how they are associated with biological processes is relevant for therapy. Okay, so how we can start how we can start, discovering those relationships, the deeper molecular and other underpinning of mutation signatures. So there's many things that some mutation signatures are associated with many processes. They may be associated with germline mutations such as BRCA. As I mentioned, they are associated with the environment, and also we can study the underpinning and perhaps a relationship of the mutation signature, but study the relationship with the changes in the molecular network and pathways. So in this talk, I'm going to explore the relationship between mutation signatures to gene expression, cell type composition, mutated gene network, and also interaction between different DNA damage and repair. I don't expect to be able to provide, to explain all of those with this great detail. So I'll mostly focus on gene expression and mutated network, and I'm going to just touch the remaining to to emphasize what sort of challenges might be hidden behind. Okay, so gene expression. So we would, there's a good reason one way to understand, what's happening, what the given process or given signature is related to, would be to study the changes in the gene expression that correlates with the strength of that signature. So to do that, we develop a very simple approach, which simply based on basic correlation. This was not the first ideas that we had, but it was the first one that actually worked. So assume that we have, for many cancer patients, we have data both for mutational data and gene expression data. So this method can be applied only to those cancers, and also would be Preferentially also would like to utilize here those studies that has whole genome sequencing so that we have a good count of those mutational processes. But in general, in this case, we have for every patient, we have gene expression for every gene. So we have this gene expression matrix by patient, and also for patients, we have the matrix that gives us a strength of the signature. So there's for every cancer type, there's a limited number of signatures that are actually present. And for each of the signature, we can compute its exposure, or sometimes I will also call it signature strength. So we have patient by signatures and patient by genes. So then we can, for every pair of gene and signature, we can compute whether or not there's correlation between a given signature and given patient. Which will give us this genes by signature matrix, for which we are going only to keep those genes which have a significant correlation with at least one signature. So after this filtering, we simply cluster those genes by the relation with signatures patterns. And once we have those clusters, we will try to interpret them by kind of basic utilizing enrichment analysis, and so that to see what are the basic processes that those clusters might be related with. So this is indeed very simple approach, and as you will see, will have some benefits, and also very quickly you will see that it has some limitations. So this is the application of this clustering technique to the breast cancer. Down here are signatures. Up here I wrote what's known about them. So the first of the signature is one of this clock-like signature. This is another clock-like signature. Those signatures here are related to ApoBec enzyme, and this is homologous recombination deficiency, and signature 8 that is not really understood. So immediately what we can see, and on the right here are the enrichment of the processes that are for those clusters of genes with provided patterns. So as you can see, the power, there are some things that there's a lot of interesting information that we gain, but there are also some limits. For example, signature 5, which is not well understood, we can really appreciate that even its clock signature is quite different from signature 1, which is the one which is better understood, which is known to be correlated with cell cycle because it's replication related. So indeed, it pops up in our analysis and correlated with cell cycle. Signature 5 is not. So they certainly, we have two signatures that tick to different clocks. We still are looking for a hint. We can see that there is there's a correlation with a bunch of genes here, and these are related to nucleotide excision repair pathways. So we have, doesn't give us quite mechanism, but at least give us some sense that this is likely to be related to some perhaps environmental processes that are repaired by this repair pathway. However, still we didn't gain any knowledge about signature 8. We know that signature, ApoBec signature, homologous recombination signature seems to be correlated with each other. We were hoping that by this analysis, we will learn which of those come first, whether this is homologous recombinations that will stimulate ApoBec pathway, but clearly there are limitations for this one. So before I will go to our next kind of try, how we are going to avoid and how we are going to resolve some of this limitation, I wanted to just show one more application of this approach. Since COVID-19, lung health is on everybody's minds. So we thought, there's so much, so one of the important mutagenic processes is smoking, right? And smoking is not, an individual that smokes, damages his or her lung, cancer or no cancer. And then we had the idea that we can use the cancer data, lung cancer data, which has quite a few control sample, and see how smoking affects, based on the signature, affects the gene expression in those control samples. So again, so this type, we are using here control samples. So we use mutational signatures that read out of mutational signatures that we get from the cancer, but we look at the gene expression from control samples. So we can learn from it very quickly is that signature 4, which is smoking signature, those are smoking related signature. Signature 1, this is the one clock-like cell cycle signature. And 213, those are apobex signatures. And perhaps as I should have mentioned that those two signatures are often seen as related to immune response. And indeed, this is what we see. We see that smoking signature is related with cytokine magnate signaling pathways. There is also another immune-related cluster that encompasses all this, more of those signatures. Yep.
In your original slide, you showed correlations between individual genes and signatures. But here, I guess each row is a gene group, or
oh, so sorry. Yeah. No, that's yeah. So this was individual, but then once we have those genes in signature here, we cluster them. So now we are those clusters of the genes that have the same pattern, right? So yeah. So those are those clusters, and that allows us to look at generic enrichment analysis. So those are so there's also interesting cluster here that is negatively correlated, which is psyllium organization. And from the previous talk, you remember that an important component of COVID and important component of many other lung diseases is the fact that there is a reprogramming of the cell types. So based on this enrichment analysis, we already can see that that there is there may be relationship decrease in ciliated cells, maybe increasing on some other cells. And what I show here are markers, known markers of different cell type in lung. This is bulk data, but thanks to significant effort in the single cell analysis for lung, we know for all this individual cell type, we know the gene expression profile. So we can use a methods that decompose bulk data into individual samples. So we did it, and then we can really see that smoking increased the amount of goblet cells. This property of smoking have been appreciated before using single cell analysis, but it was also appreciated that smoking is reducing the number of ciliated cells. And what we can see is that really the reduction of the ciliated cell is related not uniquely to smoking, but to other signatures that is often co-occurring to smoking, and a little bit also to the signature 2, which is immune related. So we get some insight also from this that may not be accessible with other methods. Nevertheless, there's only as much as we can get from clustering, as you could see. So especially remember, going back to the slide related to breast cancer, the resolution of the whole analysis was limited. So in the next, this is still unpublished. So in current work, we are trying to bypass this limitation. So what's the problem that we face is we have a lot of correlation. Everything is basically correlated with cancer growth. So what Biobatar proposed here, say, well, of course, what do you deal with too much of correlation? You try to control, you try to have partial correlation, controlling for all the confounding effects. So what he proposed here, well, let's build a network that will contain 2 types of nodes, genes and signatures. And then we know that for every genes, we have gene expression. For every genes, we have gene expression profile across the samples. For every signature, we have signature exposure across the sample. And we would like to understand the correlation between those factors when we control for all the other confounding factors. So the idea here is now to compute residual correlation between any 2 nodes while accounting for other confounding factors. Now, this is being done simple by just going ahead. You can say that at the end we are going to have partially directed network when some of the edges will have positive correlation, some of them will have negative correlation, for some of them will have some directions, for some of them we will not. So the idea is to take every individual genes and using as sparsification, so sparse sparse partial correlation technique, which will consist on the idea that for every genes, we try to find small number of genes so that the linear combination of the expression of these genes explain expression of that gene. I say expression, I mean, I should say activity because we have a mixed network of signatures and genes. There will be, so this, because of this participation, there will be many edges that will be directed into one direction because we say, okay, this gene is important to explain the expression pattern for this one, but maybe it was not picked up in the opposite direction. When you have the biodirectional edges, we additionally have the benefits that since we look at the partial correlation, so we have here the residual correlation, and we have two variables that we know that are related, but if there are no confounding factors, then often one of the statistical techniques to orient the edges is to assume that the correct direction is from the variable that is that the direction, the target variable is the one that is closer to normality. So the one that is kind of distributed is so that's additional step to help us to come with this direction. It does better. So this is random. This is we use here a database which has transcription factor target gene relationship. So it's limited. It's not relative because our network has all different type of genes, but in general, this is how it does in breast cancer and lung. Not perfect, better than by chance, and better than a simple regression tree. Okay, so but with this, so that in summary we have the edges. We have to take them a little bit with grain of salt. There are some significant positive and negative here. But now we can understand this is the BRCA network, can understand it much better. As we can clearly see now, for example, that this is homologous recombination deficiency that causes Appleback signature, not the other way around. 2 minutes? Oh my. But we could also appreciate that for the signatures that we didn't know what it does, it's now clearly related to nerve pathways. Now, we can ask additional similar questions. Now, previously we asked, how the gene expression can explain those mutational signatures. We now ask how mutated genes or networks might be associated with mutational processes. For example, we know that BRCA1 mutation causes homologous recombination deficiency. Other mutations are known to cause ERCC2 signature 5. So our next question is, can we find mutated pathways that will be associated with the signatures? For that, we utilize the method that we previously developed for drug sensitivity. When we ask similar questions, can we find mutation pathways that are associated with continuous phenotype defined by drug sensitivity? Here we replaced drug sensitivity with the strength of the given signature and ask, what are the mutated pathways that are associated with a strand of the signature treated as continuous phenotype. For example, signature 5 here, 3 here, which is known to be homologous recombination deficiency, clearly is associated with BRCA1, BRCA2, and also TP53. And also the other signatures, some of the other signatures, we have been able to find the mutations that are in some way the best subnetwork that in the best way explain this mutational processes. And I know that I'm running out of time, but I would like to just to mention just this more as an open question. We've made some progress in this, is that as this initial slide shows that what's our mutation signature, which is the end process of those of this pathway, is really related to two things. Is DNA damage, which is superimposed with DNA repair. So DNA repair deficiency interacts with DNA damage, and you can have as many different signatures as many combination of those. And we indeed have it. So for example, here are two mismatch repair signatures, very different. Are those different aspects of this mismatch repair? Or is it the case that this one mismatch repair signature is somehow occurs in the cancers that have some specific additional damage, and this is with the different ones? So this is the last explanation is the correct one. So we have to capture that. We cannot anymore use non-negative matrix factorization and assume that there is a linear combination, but we have to use the models that will be able to capture this type of interaction. So this is one thing that we develop, but there's a lot of additional questions that may be asked. Another question is, again, as we know, maybe not, okay, it was known for the biologists at least that really mutation do not hit every region of the genes with the same probability. The repair processes are developed, are emerged so that for example, mismatch repair is actively recruited to active genes so that they repair, the genes, and first, there are some mechanisms that are associated with transcription specifically. So there's lots of, so this is not all, all probability distribution fits all regions. So it's also important to understand that there will be a dependency of the mutational processes on the specific region. So that's another things that needs to be included in those methods. So just to summarize, the concept of mutational processes, the mutational signature provides a very convenient and very actually powerful way and way to thinking about those mutations, linking it to the biological processes and helping to understand standardized concept. This is some of the tools that we developed, and also another open question is how what are the different ways we can infer those mutational signatures that will be help us to capture the fact that the processes that are being involved are not necessarily additive. And with that, I would like to thank my group and funding and collaborators. Yep.
I'm particularly fascinated by your point about they don't have to be additive.
Yeah.
And because NMR just assumes that they're additive. I was wondering if, and this comes up in single cell analysis also, where NMR is becoming more popular, whereas earlier people used PCA, but now more. Now, I wonder if you can say that, say in this context, you take the log of the mutation frequency matrix rather than the mutational count matrix. And then you do PCA on that, PCA on the log count, and then you understand what are the negative values, positive values mean in the components. It's just multiplicative factors.
So, that's a good point, but what we have here is that we would like to, what's the advantage of the non-negative matrix factorization, and also our further model that it's have composition of two type signature. We call them primary and secondary. As a secondary signature act of primary, is that in the process of non-negative matrix factorization, in the process of our non-additive model, we also infer the signatures. All right, and PC will not. You will just using principal component analysis. It wouldn't provide you with those signatures at the end.
But each PC is a signature.
Oh.
Just like each matrix factor is a signature. Only these are multiplicative signatures rather than the additive signatures.
I think was a good discussion. One interesting and important fact about this work by Alexander et al. Is that you have this way of thinking about of this mutational processes, and biologists jump on it. So whenever you have new cancer data, they will decompose them into those signatures. So we now have like, not only we have the list of the signatures, we also have papers where to look, what's being known, which cancers they've been found. So this kind of, because there was lots of excitement and it still is about this subject. So we have a very quick collections of the literatures that you know, a wave that help us to interpret it. So that's one of the things. But I think your point is good. This is not the only way. Robert had another approach to infer signatures, more like machine learning type of. So this is not the autumn approach. It's very convenient because so many we have so many papers that already relate to those signatures.
So all these mutation patterns that you look at, like in the mutation is in the middle, and you look at one neighboring.
All right.
But if you increase that, you have a larger context. Are you going to better stratify some of the biological processes, or does it?
Absolutely. Absolutely. So it's not only you can increase the context, but then you don't have enough data. But it's in fact known that you know for some of the processes, like let me see this, maybe not this one, but for UV light, it's only one before matter. For ApoBec, actually, you know, there is, based on the experimental evidence, you actually know which position matter. And those would be different positions in different signatures. So that makes it a little bit more challenging. And also, you know, you have to somehow figure out that if you cannot extend this factor too much, because we will be, unless we have a much more genomic data to fill the profile. So that this is a trade-off between data and how many different mutational categories we want to use here. And Dr. Ronald, he has some work on that. Yeah.
So 96 triplet mutation types, I guess you're collapsing by complementary strand. Is there more to learn if you don't do that without strand-specific mutational? So
of course we have to collapse, because we never know which strand was the mutated one, right? So that's, but indeed, you know, this goes back to these regions, right? You know, within genomic regions, we can go transcription, not transcription. We also another thing is, you know, within say what's also important determinant of those signatures is also replication timing. And then you have leading and lagging strands. For example, ApoBec, which is known to attack single-stranded DNA. In the replication, those strands are more symmetric with the context, which one is more exposed to ApoBec mutations. So that's yet another way you will have to accommodate, and like in our approach, we just chop it into regions. Rather than having kind of, you know, data-driven, maybe with some other way of partitioning. And also, in our approach, we divided them into blocks of the replication type. And there's some tensor analysis proposed where people are trying to put it all together and put all of them, but still chopping out into regions.
Very good, but we must continue now. So, thank Teresa again. Questions on the hike, we must